Hello. Today we're going to have number 15 lecture among the series of Principles of Biochemistry by Albert Lehninger, 7th edition. Today we will do an introduction and review of nomenclature and some basic structural chemistry of nucleotides and nucleic acids. Nucleotides consist of two broad constituent groups, deoxyribonucleic acid and ribonucleic acid. The difference is the presence of a proton versus hydroxyl group on the ribose connecting moiety. We talk about nucleic acids as the sequence of or the polymer of nucleotides being the individual constituent. So when you have if you look at the polymer sequence of nucleic acids in series, evolutionarily speaking, it stores and transmits genetic information. This is a fundamental condition for life, for the ability to have energy transfers and additionally the ability to recreate itself and reproduce itself to continue its structure through succeeding generations. We talk about a gene. A gene is a DNA sequence which encodes the information for the biosynthesis of a biological product. For ribonucleic acid RNA, there are three broad categories. First is ribosomal RNA. These are complexes which facilitate protein synthesis. Second is messenger RNA. Messenger RNA is a template sequence which carries information for protein structure to a ribosome. So proteins are produced off of the messenger RNA template. And then the third broad category is transfer RNA. These are adapter molecules which carry amino acids encoded by its related sequence. So there is sequence homology between the transfer RNA and the messenger RNA. The transfer RNA carries an amino acid terminus, and that amino acid gets added sequentially to the growing protein strand. This is the biological breakdown of the three broad categories of RNA. Nucleotides have three components. There's a nitrogenous base, there's a pentose, and then one or more phosphates which are attached to it. The nitrogenous base, broadly speaking, are either purines, which is a double ring structure, or pyrimidines, which is a single ring structure. So this is the structure, and we will go ahead and highlight some things here. So three categories of purine or pyrimidine base attached to the number one carbon, and this is the pentose ribose structure here. So the presence of the phosphate group is at this point here, and the purine, so we have the conditions of the components are the base, the carbohydrate, and the phosphate ester. This purine or pyrimidine base, the pyrimidine, again, one ring, purine, two rings, and the nomenclature of the numbering system progresses 1, 2, 3, 4, 5, 6, and then 7, 8, 9, with 1, 7, 3, and 9 representing parts of the nitrogen that comprise the double ring structure. And for pyrimidine, it's 1 and 3. The nucleotide is joined covalently at the end 1 of pyrimidine and at end 9 of the purine. These are the connecting points. And the phosphate is esterified to the C5 carbon of the pentose. Purines, there are two of them, adenine and guanine. Pyrimidine, there are three. Cytosine is common to DNA and RNA. In DNA, you have thymine, and uracil is present in RNA. There's probably evolutionary permanence to having thymine in the DNA, and uracil perhaps less stable in the RNA. This is one way of describing the difference between them. We will show those shortly. In DNA, the pentose is a 2-deoxy-D-ribose, so the number 2 carbon has no oxygen, whereas RNA, the pentose, is D-ribose, and there would be a hydroxyl group at the number 2 carbon. So deoxy is at the number 2, deoxy, and then RNA is a hydroxyl group. Pentoses determine the nucleic acid structure. This governs whether DNA or RNA is produced. So the pentose structure, whether it's this one for DNA or this one for RNA, is what drives the whether you're dealing with DNA or RNA. So the difference lies here, not with the purine or pyrimidine, except for the difference between thymine and uracil here. Some minor bases are found in viruses and rare metabolic sequences. Phosphodiester bonds leak the nucleotides and they alternate in structure. So we postulate the sequence of the nucleotides, the nucleic acid rather, is a 5' and a 3' end. The 5' end lacks a nucleotide at the number 5 carbon, and the 3' end lacks a nucleotide at the 3' end. And phosphates can be present at either end. So for nomenclature purposes, for DNA we have a free, the 5' end for DNA or RNA, we have a free a phosphate ester present at number 5 carbon. That defines the 5' end. At the 3' end, you've got hydroxyl groups present at the 3' end, right? Proton here to number 2, rather a hydroxyl group at the 3' end here, because the connections are going through the 3 to 5, 3 to 5, 3 to 5. But we define the 5' end for both of these where the phosphate is. So, and then you've got in between here the phosphate ester esterified to number 3 carbon in both RNA and DNA moieties and attached to the 5'. So this is repeating sequences. The difference is at the 3', and these are important in terms of recognition in biological systems. So when we look at nomenclature, we can have, for example, P, which represents a 5' end, and then the single letter abbreviation. So adenine, cytosine, thymine, guanine, cytosine. Oligonucleotides are generally 50 or fewer nucleotides. And for the structure, we talk about purines and pyrimidines. These are aromatic molecules. They are planar. They have strong UV absorption at 260 nanometers. They are hydrophobic. The bases are stacked in aqueous solution. So the bases are to the interior, and those are hydrophobic areas, and hydrophilic carbohydrate, the backbone, is to the exterior. They're hydrogen bonds, which is the basis for interaction between the strands. This is what governs replication, what governs transcription. And adenine has 2 bonds to either uracil or thymidine, and guanine has 3 bonds with cytosine. And this is a postulate. So there are 2 hydrogen bonds present with adenine and 3 with guanine. The primary structure is a sequence of nucleotides in the polymer, again going from 5' to 3'. The secondary structure consists of the interaction of 2 chains, which is the basis for the function. So the secondary structure, for example, for DNA has 2 chains interacting with each other. And then the tertiary structures of folding of the base sequences into 3-dimensional forms. These are common for RNA, particularly transfer RNA, messenger RNA, and these are unfolded over time. DNA is also folded in tertiary structure, and these are unfolded as the biological activity commences with that structure. In the 1940s, Erwin Chargaff had rules. These predate the sequence study of DNA, or the structure of DNA. First of all, he ascertained that DNA-based compositions vary from one species to the other. But if you were looking at the same species, the DNA would have the same composition. So if you looked at the broad amounts of these nucleotides, the number of nucleotides would be the same in every animal of a particular species or every plant. And the DNA composition of a species does not change. He also determined that the amount of adenine and the number of the molar amount of adenine matched that of thymine, and guanine matched that of cytidine in all species assessed. And this obviously reflects the base pairing that occurs between those in DNA. For RNA, this would be uracil instead of adenine here, but we're talking about DNA in this case. DNA X-ray diffraction has two periodicities. There's a 3.4 angstrom periodicity in the primary structure, which is turns per base pair, and 34 angstroms. It's actually 36 angstroms for the total number of base pairs per turn. So the periodicity where a complete turn consists of about 36 angstroms. And this is the most famous photograph in biochemistry. This is from the original DNA sequence determination, and this is the helix present in X-ray diffraction, which was admittingly rather crude back in the early 50s, but sufficient enough to warrant the ascertainment of the structure. So when we look at DNA, we're looking at a right-hand double helix. It is not the sequence, the base pairings and the coil go in somewhat parallel. So it's not a symmetrical helix like you would see, say, with a double-stranded alpha helix. If you posited that, this is a beta helix, which has a certain periodicity, and again, the turn of the complete turn is 36 angstroms with 34 or 3.4 angstroms per base pair in this linear array, and this has optimum ability to unfold in the base pair, and because the biologically active form is the open form where the strand is by itself isolated and available to do transcription to RNA or other sort of metabolic alteration. So you have major and minor grooves. So there's a major groove that would occur in this, a major groove, and then minor grooves, which would be present. The minor groove occurring with the base, the individual base, and the major groove with the larger component of that. There are anti-parallel strands, so the 5' strand starting with the phosphate is opposed by another strand, so the phosphate has a homolog strand complementary with the 3' strand at the other end, and opposing that would be 3' and 5'. The primary structural stability is conferred by metal ions that coordinate the phosphate groups and is base stacking between complementary base pairs. The base replication is speculated as genetic transmission model that was deduced from the original paper on the DNA structure, that this was a genetic transmission model, and that was proven in subsequent studies. bDNA is a form that exists in nature. You can also have alpha or aDNA, which has wider periodicity, less water involved. The base is stacked on the periphery of the interior space, and this is more of a laboratory creation. It does not occur in nature. And then cDNA is another laboratory creation. It's a left-handed helix. DNA sequences are interesting. There are structural elements to it. Palindrome are sequences with identical bases read in either direction. There's a grammatical form of a palindrome. Madam, I'm Adam is one example, and you can have the same issue involved with complementary strands in the DNA sequence. You can also have these self-complementary sequences base pair with itself, so you get hairpin turns that literally look like a hairpin on the structural analysis. And you can also have more fun with DNA structure with additional hydrogen bonding, or you can have a third sequence, or you can have repeating nucleotide bases as a regulatory function to that. Originally, messenger RNA was thought to carry genetic information. It's thought that DNA is a more stable, enclosed, at least in eukaryotic systems, possibly interacting with the evolutionary transfer of mitochondria that began in bacteria and moved over to form eukaryotic components. Transcription is a process of producing messenger RNA from a DNA template. We talk about transcription, we talk about translation, which is a process of producing protein from a messenger RNA template. But messenger RNA is a molecule that it has a great deal of flexibility to it. Monocistronic RNA is where one messenger RNA produces one protein. There's also polycistronic RNA, where one messenger RNA strand produces several proteins. Messenger RNA has more nucleotide content than the complementary DNA template, and this post-transcriptional modification that occurs. These occur in terms of chemical modification of the messenger RNA additional to leader and trailer sequences. These are involved not only for cell recognition to initiate and terminate protein synthesis from the messenger RNA template, but additionally it is involved in keeping the messenger RNA segment from being digested by ribonucleases that are ubiquitous in biological systems. RNA structure: the messenger RNA is a single strand, right-handed helix with base stacking. Is uncommon and probably impossible to form double-strand helices from messenger RNA. The most common secondary structure are hairpin loops, and this is probably the extent of the messenger RNA functionality. Nucleic acid chemistry. Nucleic acid denaturation occurs with pH extremes and heat. You can get single strands. Nucleic acids are really a fairly hardy molecule to work with. You can renaturate it if you've got partial bonding still present. You can anneal the structure, renature it. You can denature it by monitoring the reduced light absorption of 260 nanometers. You can have more GC base pairs. Again, this is a 3 hydrogen bonded form, and the more GC base pairing you have, it denatures more slowly. Hydrogen bonds are pretty tenacious, they're hard to break. The double strands of RNA have greater denaturation than their DNA. As double strands go, this is an inherently less stable molecule, so DNA is a little bit better in that regard. And you can have intermediate denaturation of DNA-RNA hybrids. Usually what happens is the DNA is denatured by itself. We talk about mutations. These are permanent changes in a gene. Cytosine denaturation occurs naturally. DNA repair, it removes the uracil product and confers a genetic advantage. So one element of aging is that you lose the capacity to repair DNA, and the mutations still occur, but they occur more frequently. They tend to persist. They're not repaired. What repair occurs may not be particularly advantageous, and the more problems you have with DNA mutations, the more biological mischief occurs later on in the life cycle of an organism. So we talk about loss of base sequence mutation, or loss of purine or pyrimidine. You can also have radiation damage. The damage occurs where the radiation impinges on the surface. You get condensation of adjacent pyrimidine bases, oftentimes dimers of thymidine. You can get those pyrimidine bases transforming to cytobutadiene dimers across the same chain of bases. This freezes the base, breaks the strand, and you get a mutation at that point. You can have mutations by ionizing radiation. Those cleave the ring, the fragments covalent. These fragments become covalent structures that can bind in all sorts of myriad ways. So radiation causes about 10% of all mutations by itself. For chemical mutations, there are two broad types. There are deaminating agents such as nitric acid and nitric oxide. You can have alkylating agents where, for example, nitrosamine, the deaminates bases that's used as food preservatives in small amounts. And you can have alkylating agents where guanine methylation blocks one of the base pairs in cytosine base pairing. Oxidation damage is another way to generate mutations. You have hydroxyl radicals, superoxides, hydrogen peroxide. You can have the damage ameliorated by the host enzyme superoxide dismutase. DNA also has elaborate base repair mechanisms. There is, for example, DNA methylation. There's an S-adenosyl methionine donor molecule, and you can protect the host DNA through methylation there. It will methylate the host adenine and cytidine, and you can also use that S-adenosyl methionine for repair. In this case, this is a time-honored, a good way to repair a damaged strand. For DNA synthesis, it's generally an automated phosphoramidite method. It's analogous to peptide. You get 70 to 80 nucleotide sequences by adding DNA bases sequentially, similar to what you get with peptide synthesis. Also, polymerase chain reaction. You have DNA polymerase, a primer sequence, which goes from 5' to 3', nucleotides in solution, and a DNA segment to be amplified. This is a schematic of it, and the schematic of it takes the double-stranded component, separates it. You have a 5' and leader sequence here that's inserted. You have free nucleotide around, and then you produce an additional template with the leader trailer sequences on here. You can amplify, you can produce this initiation, then you can break this up and get additional copies. So eventually you'll be able to generate multiple copies in the polymerase reaction. This way you can amplify a single part of a genome and get multiple copies of it, and then use labeling techniques to determine if you've got a certain DNA segment or DNA genome on board. This has a great utility in analytical biochemistry and forensics and all sorts of different applications. So for PCR, we have wide applicability. You can amplify one DNA segment 1,000,000-fold. You can also apply it for anthropology, virology, genetic-based medicine, diagnostic medicine. Contamination is a problem. It's much less so than it used to be. And then another time-honored historical way is Sanger sequencing. You get the primary structure sequence. You can apply it to long chains. You can combine that the DNA sequence you're interested in is combined with a primer sequence. You can add nucleic acid analogs to stop the progression, and then you can find out what your fragment that you have after you stop the reaction. You subject it to electrophoresis, and the pattern is characteristic of the strand sequence called an autoradiogram. So when you do that, you can have, for example, a primer sequence, a template, and then nucleotides present with various additions present, and then you can apply these mixtures after you stop the reactions. You have, for example, ATP and adenine, cytosine, guanine, thymidine added to this, and you can get electrophoretic pattern that's present, and by knowing what you've added to the strand and find its relative location, you can deduce the sequence of it that way. The Human Genome Project was a situation where individual bases were determined through fluorescent labeling and electrophoretic separation of strands. So that was a technique that was used for the Human Genome Project. More recently, you can fragment and amplify host DNA sequences and immobilize them on a solid substrate, and then sequence individual product fractions. You can also use photon-generating moieties attached to the sequence. So this is an element where you can determine through chemiluminescence or phosphorescence a structure that you've added a sequence and you've attached a label to it. You can also attach dyed fluorescent markers which produce a colored light signal, and this is a highly automated step, so you can rapidly determine the sequence of, say, a viral mutation, a new viral mutation, using this sort of sequencing method to find out if you're dealing with the same material or a different one. Other nucleotide functions, they carry chemical energy, so ATP is the most readily identified one. It's a double non-carbon anhydride ester. 14 kilojoules of energy are liberated, and it shifts equilibrium to favor product formations. This is the go-to energy. Not the only one, but this is the critical one right here. There's no particular chemical preference for the adenine choice, so why is it ATP instead of something else? GTP is also used in some chemical reactions, not UTP or CTP, but ATP is the critical one. Also, nucleotides can be chemical or second messengers. Cyclic AMP is one example. It's found at the inner aspect of a membrane after ligand binding, and this messenger, this chemical messenger, cyclic AMP, initiates the next reaction in the regulatory pathway. So much of the metabolic apparatus of the organism is really dependent on the initial release of cyclic AMP upon binding of a ligand to the membrane. So these are examples of cyclic AMP here, where you get this, the ester is attached not only at the usual number 5 carbon, but also forms a moiety with the number 3 carbon here. So this is what cyclic AMP looks like. Cyclic GMP, which has opposing biological activity to cyclic AMP, has the same same structure, except that guanine is the moiety in question. And that will do it for this talk. Thank you very much.